Okay, we are live on YouTube now, and I think few participants might be joining from there. Shubhangana, can you also send a email notification maybe that we have started? Calendar invite is also fine.
Yes, Shivari, I have recently sent it.
Okay, Shubhangana and Rajni, I've given you both co-host access as well. So in case there is any moderation required in the chat box, you can do so, okay? On the YouTube, live is also there, so you can monitor there as well. Okay. Okay, then without further delay, I will start with the recording as well, and then start with the session. Okay, so hello everyone, a very warm greeting to one and all. I would like to welcome you all for today's student project session on why do brain aneurysms rupture more in smokers. And in this session, we are joined by Dijith, who is an alumni of the OmicsLogic Biomedical Data Science Research Program, who will present his research project on identification of microRNAs involved in intracranial aneurysms rupture in cigarette smoking patients. Now, before we begin, let me take a brief moment to introduce you to the team behind the program. So, we are India and US based bioinformatics and data science company with roots in India and the US. And over the years, we have had the privilege to collaborate with leading universities, research institutions, and industry partners worldwide. And our mission is to bridge the gap between data driven research and skill development. To achieve this, we work with regional and international experts who help us tailor our programs to the specific needs of students, educators, and researchers across the globe. And with the growing demand for skilled bioinformaticians and data scientists, we have developed a range of training and research offerings, including online courses, workshops, internships and fellowship, research services, as well as a degree program. Now, this is an overview of the student learning journey that is suitable for all levels. So, even if you're a beginner or an advanced learner, and for those looking to go beyond, our flagship research program provides a platform to work on real world projects with expert mentorship. I'll be covering a little bit more about this research program in my upcoming slides as well. And here's a quick glimpse into some of the courses and example projects that we cover, and on diverse range of topics from genomics, metagenomics, transcriptomics, single cell transcriptomics, as well as machine learning, R coding, Python, Linux, as well as applied example projects on different topics like neuroscience, infectious disease, agribioinformatics, cancer research, and so on. And all of these courses, they're enriched with engaging content, including visuals, infographics, and video lectures. And we also focus on experiential learning through short tutorial, practical demonstrations, as well as coding exercises. And while all of these courses, they're fully automated, you also understand that some students might require the guidance of a mentor to learn. So that is why we also conduct workshops and training programs that provide instructor-led sessions. And this also combines interactive tutorials as well as step-by-step lessons to ensure learners get best of both the theoretical as well as the practical applications. Now, here's a quick glimpse into some of the feedbacks that we have received over time. And this is the research program that I was talking about in my previous slide. So for those of you who are joining with us for the first time and not sure about it, so the research program provides a platform for the students to work alongside a mentor on a bioinformatics research project. They can choose to specialize in a specific omics field like genomics, metagenomics, transcriptomics, cheminformatics, metabolomics, and so on. And they can also choose the topic that they want to work on, like agribioinformatics, precision oncology, metabolic disorders, neuroscience, immunology, and so on. And this is the complete roadmap of how the student's learning journey is completed over the program. So on the screen, you can see the roadmap that clearly outlines the journey. It begins with students undergoing a comprehensive training in the chosen field of omics. So like I said, this could be genomics, transcriptomics, metagenomics, metabolomics, or cheminformatics. And the objective here is mainly to help the participants build a strong foundation in key skills such as data processing, analysis, and visualization. And in the next stage, students will start working on identifying an impactful research topic through regular mentor meetings as well as in-depth literature review. And in the third step, students work on data collection, analysis, and interpretation. And this is also the stage where students will start looking into data sets to identify trends, patterns, and correlations. And finally, the program culminates in students working on completing the report detailing all the analysis and interpretation they have carried out. And now let me walk you through a few examples on how students from the previous cohort have achieved their academic and career goals. So now on the screen, you can see a cohort of students from Systems Medicine graduate program who worked on machine learning projects to address various biomedical research challenges. And students have also went on to present their project in research symposium as well as poster presentation competitions. And some of the students have also gone on to publish their project work in different journals. And it is also a great way for the students to showcase their research skills when they apply for higher studies or in the job market. And it's not just students, researchers from industries also collaborate to work on different research projects. And one interesting aspect is the entrepreneurship and business innovation track. So some of the times we receive interest from participants who would like to combine science with business. So just as an example on the screen, you can see a team of industrial researchers who completed their metagenomics training and developed a microbiome test kit to improve the fertility of women in Nigeria. So these are just a quick glimpse into some of the different ways students have achieved their goals. And these are some of the preprints that have been carried out in different fields, including stem cell, space medicine, cancer research, neuroimmunology, vaccine efficacy, antimicrobial resistance, COVID-19, reproductive health, structural biology, metabolic disorders, and so on. So this was just a quick introduction that we wanted to give you, and I'll stop sharing and reshare my screen once again to take you all to the OmicsLogic platform as well once. So in case you would like to review some of these projects that have been completed by the students, you can do so. So this is the platform, omicslogic.com. And here on you, it is an easy process to create an account and look into the other students that have been, whose projects have been completed. So once you are on the website, you can click on login and you can click on create an account. And here you can enter your name, email ID, password, and you can finish setting up your profile. And in case you already have an account on the portal, simply click on login now, enter your existing email ID, password, and you can log in. So just click on this and you will be able to log in. Now, once you log in, the very first interface that you see is the course library that I was discussing in my slide as well earlier. wherein you can choose to browse through different omics field that you would like to specialize in. So there is also introductory bioinformatics if you're someone who's completely new to the field and would like to get started. There is a category for bioinformatics. You can simply click on explore and you will be able to see all of these introductory beginner friendly courses that you can explore. But apart from that, if you're interested to look into specific omics field for your training like genomics, transcriptomics, machine learning, Python, R, metagenomics, cheminformatics, you can do so as well. So just as an example, I'll open up the transcriptomics, which is also the common factor in today's webinar. And when you click on transcriptomics, you can see a list of example projects and courseworks that are related to this. So the main coursework here is the course 5 NGS transcriptomics. And as I mentioned in the slide also, all of these courses, they're enriched with detailed lessons, and all of these lessons are comprehensive and it include textual content. There are also short tutorials embedded within the lessons to enrich the student experience. There are quizzes, assignments at the end of each of the lessons as well, so that students can go through. There are also interactive tutorials, interactive materials within the lessons, so that participants can interact with certain content and further better their learning experience. You can click on load more and we'll be able to see more of the lessons. So this is just a quick introduction that I wanted to give you before we start with the student project presentation. Now I'd like to invite Dijith Rajan, who has been a part of the research program and who worked on a transcriptomics project, to please introduce himself and then discuss his project. Over to you, Dijith.
Yep. Am I audible?
Yes, you're audible.
Yep. So, hi everyone. My name is Dijith. I'm a master's student at Anhalt University in Germany, and I'll walk you through my project. Just a second. So, is my screen visible?
Yes, we can see it.
So, hi everyone. Dijith here. I'm a master's student at Anhalt University. So, my project was about identifying microRNAs involved in intracranial aneurysm rupture in cigarette smoking patients. So, this work focuses on how smoking alters small regulatory molecules in the blood and how that may contribute to aneurysm pathology. So, I come from a molecular biotechnology background with a strong interest in bioinformatics, synthetic biology, and biomanufacturing. I really enjoy combining that lab biology with computational analysis, and this project reflects that integration. So, the main objective of this study was to compare microRNA profiles across three groups: smoking intracranial aneurysm patients, non smoking intracranial aneurysm patients, healthy controls. So, this will help us to understand and identify the smoking specific microRNA signature and understand the affected biological pathways as well. So, in simple terms, this study asks one question: how does smoking change small regulatory molecules in the blood of a patient who already have the brain aneurysms? For that, we need to understand what is intracranial aneurysm and what is microRNAs. These are two important part of the puzzles. So, our blood vessels carry oxygen and nutrients to the brain, and sometimes a weak spot forms in the vessels, and this bulges outwards and balloons, and this forms aneurysm. It's visible on the right hand side of your the slide. And why is it important? Because most of the aneurysm are highly it has high mortality rate and long-term disability, and it also is silent, and also it's very hard to predict when it will rupture. And when it ruptures, it can cause hemorrhage. So that's why it's very important. So there is no one causal agent behind it. It's an inculcation of different factors. So a few of them are listed here. It's one is genetics, and another is vessel wall biology. Then smoking, of course, is one of the major factor because it's strongly associated with inflammation, vessel wall damage, breakdown of extracellular matrix. And smoking is a major risk factor here because it accelerates all these processes. And yeah. So microRNAs are very small non-coding RNA molecules. So they are gene regulators, and to be honest, you can think of them like volume knobs, but for genes here. They are stable in blood plasma, which makes them more attractive as potential biomarkers. And they play key roles in inflammation, vessel wall biology, and the remodeling of extracellular matrix. So we used publicly available plasma microRNA expression data, which were obtained from GeoDatabase. And in this study, samples were divided into three groups: control, the healthy controls, and uh, aneurysm patients with non-smoking, who are non-smokers, and smokers who are aneurysm patients. And each group had 10 people to them. And while it's known that microRNA are altered in intracranial aneurysm, and also it's generally known that smoking also alters microRNA in general. But the gap this study is trying to address is how smoking changes microRNAs, specifically in IA. That's still unclear, and that's the gap this study is trying to address. So this slide summarizes the overall analysis of workflow. We began with publicly available data from geodataset, and in that the samples were divided into three groups. So first we prepared the data and did the exploratory data analysis, and during that time the filtering, the normalization, dimensionality reduction was done. After that we conducted comparative and functional analysis, and the differential expression analysis were performed using DE-seq2 to identify significantly altered microRNAs across three samples or the comparisons. And to understand the biological relevance of this data, we carried out cake pathway enrichment analysis using both Enricher and ShinyGo. And after that, protein-protein interaction network analysis was performed to understand the hub genes, and it was done in Cytoscape to identify the hub genes, and it was followed by the integrative interpretation of the results. So together, this pipeline allowed us to move from the raw sequencing data to biologically meaningful, relevant interpretation and the insights. So after normalization, principal component analysis was performed to explore the global expression patterns. So here the PCA reduces thousands of microRNA into two dimensions, so we can visualize the clustering and the separation. So we can see clear separation between groups, indicating meaningful biological references or interferences. So the differential expression analysis revealed a strong effect of smoking, and volcano plots for each comparison illustrated both the magnitude and the statistical significance of it. So in smokers, a large number of microRNAs are downregulated compared to the upregulated, suggesting a suppressive regulatory effect of smoking. So after that, we did the heat map analysis, and the heat maps provide a final visual confirmation for distinct transcriptomical profiles, which is associated with each conditions here. And we have clear separation among all of them. Specifically, we understand the clear separation between smoking and non-smoking. From that, we can understand there is a specific microRNA signature that's attributable to smoking. And from Venn diagram, we understood there is an overlap between comparisons. The analysis identified around 84 core microRNAs that were differentially expressed across all the three comparisons, which shows the presence of conserved IA pathology and associated signature of microRNA. And this can be used as a potential universal biomarker because relevant of which condition that is, it's present in everywhere. So it can be considered as a potential universal biomarker. So this table highlights the most strongly upregulated and downregulated microRNAs based on fold change and statistical significance. And these microRNAs were used as the candidates for the downstream functional and network analysis study. So because individual microRNAs are very difficult to interpret biologically, so pathway analysis, enrichment analysis was performed, first using enricher, and TGF beta signaling pathway was the most enriched one, followed by AGE rage signaling pathway, then focal adhesion, Cushing syndrome pathways in cancer, and we did use the same data and did the analysis and the enrichment analysis in ShinyGo to reduce tools based biases, and we got almost a similar result where TGF beta signaling pathway was the most enriched one, followed by AGE rage and ECM receptor interaction, adherence junction, so on and so forth. And across the two platforms, the top enriched pathways are shown here. And one interesting thing that we found out was that all these pathways are known to associated with blood vessel structure and inflammation. So the network analysis was used to identify hub genes, which act as control points. So key hub genes include here IL-6 for inflammation, FN1 for extracellular matrix organization, PTEN for PI3-AKT pathways, EP300 and HDAC1 for epigenetic regulations. So importantly, all of these genes are associated or established roles in vascular biology or aneurysm related processes. So, to summarize, intracranial aneurysm significantly, not slightly, alters circulating microRNA profiles, and smoking does amplify this by having a dysregulator or a suppressive effect on microRNAs. That's the microRNA signature that we got here. And critical vascular and inflammatory pathways are affected here. And this finding helps us to understand molecular insights into how smoking associated IA is working, and also helps us to identify the candidate biomarker and the therapeutic target for the future studies. So, just like any studies, this one also have limitations because this one, the primary one is that it was a computationally done validated analysis. So, and the sample size were very modest and the design was very cross-sectional. So in the future we need to have experimental validation, and the study need to be a longitudinal study across a period of time to help us understand what's happening across a period of time. So that's about it. I would like to thank everyone for bearing with me for this time, and thank the OmicsLogic team as well. And I would like to thank my mentor who has been supportive along my journey. And to talk about my journey, I was a complete noob starting out in October. To be honest, I had no clue about the uh, side of omics, like how it works. I have heard about omics, transcriptomics, proteomics, and everything, but I haven't delved deep into it. I just had like a base idea, so I just took that faith of leap. And when I looked around in net, like where I can study the relevant skills to become a bioinformatician, especially on the omics side of things, that's when I found out OmicsLogic, and I reached out to them. So it was like a very positive experience for me along the 3 months of time, to be honest. And the project I worked on was like also very interesting for me because I did have formerly a neuroscience background. So yeah, so if anyone is on the verge of deciding like should I join the program or not to upskill yourself, I would suggest you to take that leap of faith and go for it, because I was in your same place 4 months before. So yeah. Thank you. That's about it.
Thank you so much, Dijith, for your presentation. If the participants have any questions, you can post in the chat box. But I can see more participants had joined after we had started with the presentation. So I'll just give a quick overview of what we have discussed so far, a summarized version. Dijith, can I share my screen next?
Yeah, sure.
Okay, thank you. Okay, so for those participants who have joined with us just now, so we were discussing a student project presentation by Dijith Rajan, who was part of the OmicsLogic Data Science Research Program. And for those of you who are new to the program and would like to learn more about it, so this is a program that provides a platform for the participants to work under the guidance of a mentor on a bioinformatics research project and complete a research project as a part of their dissertation or for their end semester project, or it could be just to simply gain some research experience. It could be also for participants who are interested to work on preprints and publications as well. And the participants can choose the omics field that they want to specialize in. That is, it could be genomics, metagenomics, transcriptomics, cheminformatics, or metabolomics. Initially, the students will undergo training in one of these omics field, and they can choose a topic that they want to work on for their project, such as oncology or infectious disease or astrobiology or neuroscience and so on. So right now we had a student presentation by Dijith Rajan, who had completed a transcriptomics project. And all of these student projects are also available on the OmicsLogic platform. I'll quickly share my screen and show you where you can find it. If you're already familiar with the platform, then you might have an account on the platform as well. But for those of you who don't have an account, you can simply create an account here. I'm sharing the link in the chat box once so that you can create an account here. And once you create an account, you can head over to this project section and see all of the projects that have been completed by other students, just like Dijith, but in different interesting projects as well, just like Dijith's project on like cancer research, there is neuroscience, there is space medicine related projects. There are also different projects on machine learning. So all of these, you can have a look into it. And we also get a lot of queries from participants who are interested to work on a research project, just like Dijith. And so if you're interested to work on a research project similarly, we also have some ongoing research projects that you might be interested to work on. I'll just quickly show you on screen. So this is a transcriptomics project that we have currently ongoing. So if anyone is interested to enroll in this project, you can get in touch with us. And just at the end of my slide, I'll launch a poll. So you can fill out the details there and let us know how you would like to connect with the team. And another project is a 3-month project on virtual screening for drug target discovery. And we also have another project on multi-omic analysis in cancer, which is a 3-month project. And we also have another project on a 2 to 3 month project on machine learning for identification and validation of predictive biomarkers. And finally, another project on immunoinformatics, which is also a 3-month project. So if anyone is interested to work on a similar project, I'm now launching a poll on the screen. You might now be able to see a poll that has popped up on the screen. I'll quickly read out the fields as well, so that in case you're not able to read it, maybe you are joining from phone also, it will be easy for you. So the first section is full name. You can add your full name. And also there is a second question is on email address. The third is on which area of research you're interested in working on. Just a second. Yes. So I was saying the third question is on which area of research you're interested in working on. So there are different topics that have been listed, like precision oncology, CAR T-cell therapy, infectious disease, machine learning, metabolic disorders, drug discovery, neurodegenerative and cognitive disorders. And there is also an option at the end called others. So let's say you're interested in working on a bioinformatics projects, but that is not listed here. Then you can select the others option. And in the fourth section, there is an option for you to fill out more details about what this other option is. So you can simply fill out the poll here. We will keep the poll open for just few more minutes. And for those participants who would like to learn more about the research program, you can simply fill out this poll and our team will connect with you. Okay. Now I'll also share with you this scholarship form. Shubhangna, can you share the link in the chat box for the scholarship form? Yes. So on the chat box, we have also shared with you a scholarship form that you can fill out if you're interested to apply for a scholarship when you enroll in the program. I'll also quickly read out the fields for this. So there is email. You can make sure that you enter a proper email and a full name that you want to connect with, and also a phone number. Also mention the country, company, organization, or university you can mention, and also the level of education. Then you can also mention if you have a prior background in research, yes or no, and which research path you would like to pursue. Is it independent or group project? And if it is a group project that you're interested in working on, you can also mention the number of groups, the number of students you would like to work with, and also the project duration. And finally, what is the motivation for undertaking this research project? Is it for dissertation, publication, research experience you can mention? And also, you can mention few lines about why we should consider you for scholarship. At the end, there is also an option to upload a CV. So if you have an updated CV, you can upload it here. So we'll keep this form active. Shubhanga, till when will this form be active? Shubhanga? We cannot hear you yet. Shubhangna, are you there on call? Or Rajini, one of you?
Yeah. Hi, Shigori. So we can keep it open because I can still see people are joining. For maybe 5 minutes more, we can keep it open.
Okay, sure. So the scholarship form, then we will keep it active maybe for 24 hours and you can fill it out. We'll keep the poll open for a few more minutes so that participants can fill out. Yes, Dr. Jimmy, you have any questions? Please go ahead.
Sending the link in the participation messages itself.
Sorry, I didn't get your question.
Will you be sending the link in the message chat box itself, or will it come as a email?
Yes, we have already shared it in the chat box. I'm sharing it once again, but we'll also share it over email as well once, just so that you can keep a track of it. if you can just check the chat box, I've shared the link. The poll, however, we will close it in just few minutes. You're waiting for other participants to fill it out. Emanuela is also asking for scholarship link. I've shared the link once again. I hope you're able to access it now.
Yes, I can see now.
Perfect. Thank you so much, Emanuela. Okay, I can see participants are still filling out the poll, so I'm still keeping it active. Any questions, if there are related to the program, you can connect with us and our team will be happy to assist you. Okay, I'm closing the poll in a few more minutes. Okay. I'm ending the poll. Anyone, would you like me to pause? Please let me know. If there are any questions for the student speaker, you can also let us know for that as well in the chat box. Okay, I guess that will be all. I'm ending the poll. Okay, we have also shared our contact details in the chat box for those of you who would like to connect with a team member and get more details about the program. You can connect with any of our team members on the numbers that has been shared in the chat box. They're also available on WhatsApp. So if you have WhatsApp, you can drop a message there. But if you would like to connect with over email, our team will also share the email ID in the chat box. If there's any question related to how the program is conducted or what will be the structure, what will be the schedule with the mentor sessions, what will be the program pricing, so on, you can drop a message and our team will connect with you. And once the webinar is over, we will also email you the recording. So in case you joined the session late or would like to rewatch the recording once again, you can do so. And we'll also share with you the scholarship form link. It will be kept active for 24 hours. So please make sure that you fill out the form till then. So there is a question from Dr. Jimmy, criteria on getting the scholarship. Is it full scholarship or partial? Rajni or Shubhagna, are you able to answer that?
Yeah, so the scholarship will be partial. It is customized depending on the condition or depending on the participant to participant. So we'll check with the people and we'll discuss what is the condition and why they need their scholarship, and then we will offer you the whatever the discount we can offer.
Thank you so much. Okay, then. So thank you so much once again, Dijith, for joining with us today and sharing your project presentation. And thank you to all the participants for joining with us today and listening and being attentive. And once again, If you have any questions, please feel free to connect with us. We're looking forward to seeing you in some of the upcoming events. Thank you and have a great day.
Bye.
Thank
Bye.
you.
Thank you.